G'day, good morning, good evening. I hope you're well and doing fine where you stand or sit and listen. This is a medicinal cannabis program where it respects the law and I only promote organic growing. Synthetics or chemicals can be added for foliar maybe, but yeah, anyway, don't want to get into it. Today is a open topic, so nothing's being planned or organised. So I hope that there's some questions or stuff on your mind that you'd like to clarify, and I can show some slides from my university studies and we can somehow make the topic a little bit clearer. Yes. Hello, Dave. How you going, mate? Dave's not happy. He doesn't like the winter. Yes, it's starting to get a bit cold down here. It's lovely. I love it personally. And plants like it too. So the cannabis plant, it can tolerate it. You can get ones that tolerate it a bit better. Actually, I've got some of the snow with some snow on it. But they don't like it for long because it kills things. It slows down enzyme processes. It's really bad for the microbe interactions. It's not good. All of the xylem and the phloem in the plant, that really, really slows up because it can't get the processes it wants to make it flow faster. So it really sort of nearly halts things. So it's not a good, I'll wait for a few stuff to come back. I'll get one of those slides of it. Not a slide, a picture. Microbes, eh? Yes. I haven't talked about microbes for a bit. Microbes might be all right. There's one. I see a photo. Share the screen. Entire screen. Why not? This photo. This is a bit of frosty fun. What is it? GGC. God's green crack. Yeah, a little bit of snow on it. It's all right. It'll handle it. Actually, there's another one too. Anyway, don't want to start with the snow action. See if there's any stuff any of you people wanted to mention or talk about, and then I can start and go that way in the stream rather than me just starting to share, because I like the microbes. I saw that earlier. Maybe talk about microbes. Hey, Ned Kelly, what do you think? Morning, Ned Kelly. Yeah, which way do you want the show to go today? What's on people's minds? I like the microbe thing because I just saw that before when I was looking for that snow pic. That's the only reason. Where's the other snow pic too? Where does it end up? It's under that live section. Lives. Yes, I'll get back into that too. Where's that snow peak? I saw it before. Hang on. Bear with me, please. Are you kidding? Oh, there it is. Another snow peak. So the last one before that was from BC in Canada. And this one here is from Alaska. There's a plant grown in Alaska. I lived 2 seasons up in there in the Yukon. It's a wonderful place. I would love to go back. Nice temperatures too. That's where it gets cold. The car tires froze at minus 38 Celsius, which is virtually the same as Fahrenheit. That's why I knew it was at that temperature, because it was minus 38. The car tires would nearly go do. But yeah, there's some frosty fun. So see how it's on the top there? So it doesn't want to be that, because that's going to chill the substrate real fast. I'd remove it from that and give it a shake, and it'd be right. It'd be right. It's not going to like the temperatures, but it can withstand it for a few days at least. But while it's got packed on it, it's stopping a few processes. Hey, good morning to, good evening to J-Mac over there in Oregon. And Birds, how's it going, Birds? If I'm going to start and talk about, what was the name of the frosty plant, says Dave. That was God's green crack. Yes, that was beautiful to smoke. Yes, that was that one. So if there's no real suggestions on any topics or anything that you want to clarify, I'm going to start talking about some microbes, I reckon, because that came up before when I was looking for that photo of the cold, just because it's starting to get cold down here in Australia. Snow on the mountains, getting down to negative at night, just below zero temperatures, 34 Fahrenheit. It's very nice. I like it. That would do okay here in the winter. Yes, you can, even if you get genetics you can climatize them or make them go in colder and hotter regions, they can tolerate those temperatures. It's still going to slow down the processes inside of the plant. So you won't get the yield. That's why the Ruderalis is from Russia. That's where it's that species of plant. And that's generally, it doesn't grow more than a foot tall, because for that reason, it struggles through the summertime or the springtime. Yeah, righto. Well, I'm going to start and talk about some microbes and show some microbe stuff. What do you reckon? All right, microbes. I'll share screen. You can see all the microbe stuff. It's not microbes. That's all the lives. I'll get back into doing this. All of the updates on the latest topics in medical cannabis. I'll start doing that. I'll do that next week, actually. How's that? Where I'm sharing all these rad slides from all the latest studies and things like that happen, so you can keep up on what's happening in society. What's something cool to show here? Education. All right. I should look for a microbe thing. Education. If we need science to legalize cannabis, can we please see the science that was used to make it illegal in the first place? Actually, I don't want to sway the show that way, but it's because it was, there's another slide here that explains that. No, I'm not going to go through them. We'll get back into the microbe stuff, unless there's been Buds. What's the name? No. Lene, good morning, Lene. How are you? Top of the morning. Blinky, what's happening, Blinky? Microbes, I think that's all I can think of to talk about. And next week I'll get back into doing those lives. So I'll do some. So that means next week will be on plant genetics. Finally, I'll have to get on to Frank and see if it suits Frank too, because he was into the mutant genetics, and I'd like to discuss some stuff with him because he's a nice fella. So I'll see if he's available, and if he is, I'll do that next week. For this week though, we want the microbes. So I'll go back to the microbes folder. Here we go, microbes, and back to your screen to share screen. Now we're going to get into some microbes related to medical cannabis and see how they, actually that is quite related, because there's a topic coming up in plant science on the frontiers of plant science, I think, but it's a free lectures that's being given online mid-June and it's related to the plant-microbe relationship. So this will go kind of a bit with that, I suppose. And look, this is the microbe stuff. This is cool. Oh, this doesn't show the plant-microbe relationship. This is a plant. This is the biology relevant to microbes, but more so bacteria, and what is their elemental breakdown. So you can see up here, the majority, which is essential for all life, will be in this goldy color. So the hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur. So those ones are all related to all microbes. And then you go down for the major anions and major cations in these next few colors, which is pretty much half of them. And then it goes down to the trace elements and different ones that are specific to breaking those down. It's more so in the metals and in the alcohols, alcohols, and the anaerobic ones. I'm going to stop sharing. Availability of nutrients. Hang on, where was it going in? Sorry. I just liked that before because I haven't seen that very often. The actual elemental breakdown, the NPKs, you could call it, of bacteria. So that's why sludge is beneficial because it's got a lot of this stuff in it. Availability, no. I'm a bit lost here, sorry. I want to go here into this folder because we're just going to talk about some microbe stuff, and this is my microbes folder and the association, the relationship between plants. Oh, you can do the rhizophagy. That's the relationship between the roots and the plants, because rhizos is root in Latin. Phagy means eat, so the bacteria around it. Let's go through that first. Suppose. Let's just go through the PDF. So this is how plants and soils, microbes, relationships in the rhizophagy. To summarize it, it's how microbes interact with roots. So there's going to be a lot of these root tips that are going to be shown here, and you can see inside of them through stains on how they move and how they interact throughout the tissue, and then what different relationships at the end. It'll show some experiments where if you have them in, the difference between having them and not having them, and then it'll show the nutritional breakdown on the benefits that the plant gains from having these bacteria or endophytes inside of them. So what's an endophyte? So endophyte, endo means inside, and phyte means phytes. No, that's phagy. I think phagy means eat. No, I'm not sure what phytes in Latin is. But the endophytes are the microbes that are inside. So endophytes can live in the roots or in any type of tissue, but this is more related to the roots. Bacteria, endophytes, and seedlings in hemp. So this is them inside here, the little round dots. I'll zoom in then. Over here. Now it's too blue and big. That'll do. So I'll skim over this. Bacteria and root hairs showing recently divided pairs. So it helps with, this is the primary root. So you've got the big long one, and then you've got there how they start, and they have secondary roots, which is the lateral roots that form out the sides. So it helps with formation of those. That's what this is proving. Where do these endophyte microbes come from? Well, they come from the soil. They come from plants, fish microbes out of the soil and put them on seeds for seedlings. That's why you can get different startings of seedlings or seeds, their germination process. If they've been produced in a sterile condition, like in a hydroponics place indoors where they don't use any types of bacteria or fungi or beneficials or anything, you'll have plants might struggle, or the seedlings might struggle, because they're picking up whatever the new microbes can culture and get onto it from the local material that you're using. But if you source them from an organic breeder, they will already have all the microbes inside of them, or these endophytes, and that will help the seedlings germinate and start quite fast and a lot better. This is why it is important to have healthy and diverse microbial communities. Yes, in your substrates. This isn't just to do with soils. That means, you can say substrates. Do plants consume bacteria to obtain nutrients? Yes. Do plants consume bacteria to obtain nutrients? Well, the endophytes inside of them, they have to go through their cycle, and whatever their cycles are, they will be releasing whatever their processes are. Like nitrogen-fixing bacteria will change the element from one element to another element. So that might be what's going on inside of the tissue as well. So they do work, and they're symbiotic. So if the plant doesn't recognize it or doesn't work with it, it will try and expel it, like they do on the leaves. You get the hypersensitive reaction when it means if there's a little dot there and it starts to burn around it and die and go outwards and die further out. That's a hypersensitive reaction, because whatever landed there or got into that tissue, the plant wasn't symbiotic with it, and it wanted to kill it. So to kill it, it kills its own tissue, as an example. I've been going on about this for a while. I'll stop here for a sec and see if there's any questions. We're just about ripping right into it. Hola. I do need to be scholar in curing properly. Curing, hey? Maybe I'll get onto that after that, talking about the Rhizophagy scale, all these microbes, plant relationships. Supreme Great, good afternoon. How are you? So back to plant microbe relationships. Which was this one? So this one shows how they come in through the root tips, the rams, the root apical meristem, and it proceeds through the cell tissue, and it comes out through the root hairs. And then it goes round through the soil, where microbes recharge with nutrients in the rhizosphere. And then the microbes enter the roots, actually in the periplasmic spaces, and then they carry the nutrients from the soil back through it. And the nutrients are exchanged from microbes by reactive oxygen produced by nitric oxide from root cell plasma membranes. So that's pretty cool. Actually, here's a stain where you can see that they're stained, and you can see that the microbes are stained with blue. So you can see their pathway, how they've come in through the tip here and they're out exiting through the side. Now what? Oh, hang on. It's not as, I was going through the PDF. Sorry, I'll get the PDF back up. Try screen. Here we go. No. Oh, what's where I was up to? Very good. Oh. So bacteria, they enter the root epidermal cells and colonize the root tip. You can see how it's stained in here in blue. That's then inside of it. This is actually inside of just one cell. So this is a root cell wall, is the grey bit, and inside of it here is the cell. So it's you can see the bacterium in the sides. So they've actually gone inside of the cell wall, and they sit just in beneath the cell wall inside of the cytoplasm there, in the periplasmic spaces, doing their bit. So the rhizophagy cycle, Microbes modulate development of seedlings. The microbes trigger the gravitropic response. They trigger root hair elongation. They increase root branching and increase root and shoot elongation. So pretty much you get a bit all over benefit from it. Microbes rule. It's good. This Bermuda grass seedling, actually I don't want to talk about it because it's got nothing to do with medical cannabis, but it just goes to show it. So that's, you can see how it's colonized inside of it, and it started to produce these root hairs quite well. And the primary root and the secondary roots are forming very nicely and fast. Some Pseudomonas species are working particularly well, and with medical cannabis, Pseudomonas petita, and there's a few other Pseudomonas species that work really well too. They're very beneficial that I particularly use. Why is root hair growth linked to the presence of intracellular bacteria? So why is it? It's because they put out different types. Here's Pseudomonas fluorescens, which is also very good in medical cannabis. It puts out different types of hormones. Oh, here you go. Ethylene. And ethylene triggers a little bit of root growth hair. So just inside of these Pseudomonas fluorescens, which you can get out of one of the leading persons on the market, look at your beneficial bacteria, turn it over, and you'll see that on the can or whatever it is, or in the little plastic jar, they've got the breakdowns on what microbes there are inside of it. And there's one particular one that has maybe 20 or 30 inside of it, and it has Pseudomonas fluorescens. So by doing that and putting that in the early stages, you're going to trigger a lot of lateral root hairs because of the hormone ethylene. And there's not enough ethylene to give it a triple response, because if you know you've got too much ethylene, you get a triple response in the seedlings, and you'll see there's a J hook with the root. It'll try and curl up, so it won't have the gravitropic response. I'm trying to go with the gravity. It'll hook back around, and you don't get that with that. That's the molecular breakdown of the microbial ethylene root hair exchange. There's a stain just to show that they're stained with ammonium molybdate. So it's the blue and purple color. You just see a little bit of it going on there, and see up through there. So it's coming in through there, along through there, and then exits out through there. So it doesn't go anywhere down through here. And even in here, if you zoom right in, you can see the little bit of purple. So that's that's one cell. So inside of this cell here is the microbes, and you can see the purples, that the stain shows purple or blue if there's an emission of the ethylene. Blue arrows outside the root hairs also stain ethylene using ammonium molybdate. Okay. Whatever bacteria are evident in root hairs, ethylene appears to be produced. So it doesn't just have to be Pseudomonas fluorescens. If you work for some really good endophytes, you can see what sort of endophytes your cultivars work with by doing when you do a harvest, you would cut the stem and dry the stem right out, like when you're drying out the leaves, and not drying out the buds, sorry. And then once it's very dry, you'll put it back in humid conditions. So leave it in like a jar or a bag that's above 85% humidity. So you're giving them the right conditions for it to germinate in, and then you'll see out of the end of the stems will be little spores, little round seeds. There's some photos of them. All right, I'll go back and show some. I'm still sharing. Yes, I am. I know some down here. Is that some? Ah, that's a culture. Oh, no. Fungi, maybe. There's some cysts. That's not the stem experiment, though. That's the spores they produce. So you'll see them sitting outside of the end of the stems. And then that's the germination. And then once you put them in the right conditions, above whatever conditions it needs, the water activity level, possibly you can guarantee if it's above 85%, they'll germinate, or 90%. So whack them back in that conditions, and then they'll germinate, or put them on a culture plate, and you can see what endophytes you have inside that are beneficial or symbiotic with your particular cultivars. Right, I wonder if anyone's got any questions on that, and then I'll get back to that PDF. Yes, all right, I'll stop sharing. Add that one to the stream. Linky, no worries. Jimmy, how are you going, Jimmy? John 420, Grove bags are great for curing. Oh, yeah, good stuff. You can, talk a bit in the chat if you want and help Linky out. Very good. They are. The microbes, get back to this. Wow. That's a PDF. Okay. Root hair showing microbes circulating along the interior of the root hair. All right. Let's sustain. Let's go into something a bit better. Go through the pores. What happens to plants without? Here we go into some experiments. What happens to plants without any endophytes inside of them? So you can see on the right-hand side there, this hasn't got any inside of it. So it's just it's germinating under sterile conditions. And this one here on the left, it has endophytes inside of it, particularly Pseudomonas species. Isolated from Phagamites australis. Wonder where that was developed or found. Rice experiment. Pseudomonas. This is another one too. You can just see the development of the seedling. So it's very poor on the right, none. And the nutritional absorption function that happens with the microbes. So on the left, there's a definite increase, and on the right, they're sort of struggling a bit, you can see. So on the right, the proteins have been reduced from not having the microbe interaction. Seedlings with bacteria absorb 30% more 15 nanotubes isotope label nitrogen than seedlings that lack bacteria. So they have a better start as well. There's some really good tests about that. This might be good about talking about nanotechnology, jumping into that. So no bacterial control. Or on the right, this is Bacillus species. Bacillus subtilis is very symbiotic with medical cannabis. So you can see there's quite a good difference. And look at that. Not only that though, see how they're a bit wimpy? Look at this, it's a bit stronger, quite strong, like just natural, I suppose you call it. This, you're going, why are they struggling? Well, it's only because they've just got no bacteria. There's no deficiencies. It's just the bacteria. It's the only difference. Now we get into the elemental breakdown and see if there's any benefits from having no bacteria to having bacteria. And this first one shows nitrogen. So on a dry weight plant tissue, so they would have dried out the tissue, the green leaf material, and then put it under a nitrogen test. And all these are different types of chemical tests to show the difference of having non-bacteria to bacteria endophytes. So you can see there's what, 0.3 to 0.5, so it's nearly double the benefit from bacteria, from nitrogen. Phosphorus, this would change too, depending on the microbes that you have inside of it, but just in general, this is it's the difference that you'll get. So it's changed from a 13 to a 17 from having bacteria, so it's gone up a bit. Potassium, it's massive difference. So from 0.5 up to 1.6, you could say. So that's nearly a triple response. So if you've got a potassium pig or a potassium deficient cultivar that shows before anything else, you can know that if you put in some endophytes, there's a high chance that you will eliminate that potassium deficiency and let the endophytes work for you. Calcium, marginal difference, 0.35 to 0.45. Sulfur, etc. Manganese, magnesium. So it's quite a lot. So the overall, there's the three benefits, you could say, from having endophytes inside. And it's plants absorb nutrients from the microbes. There's a second benefit would be increased oxidative stress tolerance in plants because they increase reactive oxygen species. So it works way more better for the plant. So it's a lot in less stress too for the plant by producing them. And the soil fungal pathogens have reduced virulence. Soil fungi drained of nutrients by the Rhizophagy microbes. So that means that they're not going to be as proliferant. So in medical cannabis, we've got the Mycetes, which they're the water-bound pathogens like Pythium, Fusarium, those awful ones. So by having these endophytes, it will help with that as well. There you go. And there's some references just for the persons that wrote this. Very good. Stop sharing. Righto. Any questions? Dave says, I was just looking at group bags on eBay. Oh, yeah. All right. Well, maybe we can talk a bit about curing, I think. What was it? Post-harvest productions. Yes, curing. So post-harvest productions of medical cannabis. Another stage which has to be mastered. And it's forgotten about too much, sadly. What can I show from that? Curing. Post-harvest productions. Let me think. Let me just go through a few slides and see if you've got any questions out there. And I'll see what I can find on that. Ploidy curing. So what would that be with temperatures? It'd be secondary metabolites. Maybe secondary metabolites, because there might be some tests on some different temperatures at different. No, I got something on it because I've talked about it before. Just a matter of where it is. It's not really in my face. There's no real curing thing. Actually, I did a video on that, didn't I? Because that would be a lot of references under that. I did actually. I did a few on the. What's it called? Sorry, I'm trying to talk and do that at the same time. Holy mackerel. No. Drying, that's what I was thinking. Here we go, bids made. Drying cannabis to perfection. Oh, really? I've only got the vids. I haven't got the slides. There's another drying one I've done then. How to fast dry within 36 hours. Oh, dead set. It's only a video as well, and I mustn't have shown any slides. Oh, wow. So it's a fast drying. It's got nothing to do with curing. Well, it's with curing. It's got to be to the right temperature. There's got to be something I can show. Hang on, I'll just get something so I can talk to with cannabis in the background. How to fast dry, enhanced drying, cutting birds, leaves and essence. Just going through kind of slides. Oh, yeah. Let's have something in here. Post-harvest. Oh, there was a good one. Rad. All right. I've got one. I found one that's applicable. So for curing or post-harvest operations of medical cannabis. So the environmental factors really affect and deteriorate cannabis, and that's why we dry it, because this is cool as. Well, relative humidity has a direct effect on the growth of the microbes in association with temperature. So if you can control the humidity, you've basically half won the battle, because they need an activity water level for them to germinate or sprout. And the combination of temperature and relative humidity can alleviate or reduce the deterioration dramatically. So if you're going to, you should always have a humidity meter, more so than a temperature meter for your drying, because when it goes after drying, you put it in the cure. And if you dried it properly, curing should be straightforward. None of this burping the jars, none of this, that sort of thing. You don't need that if you've dried it properly. It just goes in, seal the bag, lock it, and it's done. Then when you open it, it should smell like it does when the plant was growing. So no off smells of the terpene profiles or flavonoid profiles mixing between each other, because the temperatures wasn't dried properly, and the humidity still remains there and allows the processes chemically to happen. Like here, what's this one? The moisture content. So if you've got moisture content on this side and the relative humidity is high, so this shows down here, microbes or insects don't grow if it's below 35% humidity or moisture content. We're above 65, you've got fungal and insects starting to grow, and then bacteria is all above 90. Like the lowest is 65, which is Saccharomyces cerevisiae, is the yeast, and that starts to germinate. So if you've got those spores like I showed before, if you've got these sitting here and then your humidity gets 265, like down, right down the bottom, see how they got little trichome-like things on them? They're probably starting, oh no, it's not. No, it's not germinating. But 65 anyway, that's what that chart's showing. And yeah, the temperature, see how it doesn't have much of a great effect to what the colouring is above. So it's more so the humidity when you're drying it. So if you can dry it out properly and make sure, if you've got big buds, it's not going to be just because the outside's dry, the inside won't be dry. So that makes it very hard to cure or dry big buds. So a meter would be good for that. One of those wood moisture meters, and you jab it into the sides of the bud, and you can see what moisture you've got into it. And then you can put it in at different levels, and then you'll know how much moisture is actually in it at different stages. And then that'll teach you to know the weight of the buds and that type of thing and what you're getting. And if you push them out, you squeeze them in, how fast they expand out, or do they expand out? This bar graph, this float bar graph here, sorry, relative deterioration rate. So you've got lipids and enzymes. They also break down. This is the water activity level. So you could say that's like nearly like the humidity. So 60%, 65%, you can see here. This is where the Saccharomyces starts to germinate. And then as it gets up to above 90, all the bacteria start, and some start at 85 bacteria. But enzymes, they start to break down and chemical reactions happen, which isn't healthy. So if you want to get the best out of your medical cannabis, there is ways to fast dry it and maintain the stability of things, but it's all to do with temperature if you're fast drying it. I got a video on fast drying, a couple of them. The post-harvest diseases are most severe when relative humidity is 90% or higher. So what you do is, after you've harvested, wherever you've hang dry, hopefully, you would have a humidity meter, and if it gets above 65, you need to evacuate the airspace or raise the temperature, because temperature is relative to humidity. So if you raise the temperature, the humidity will go down. But keeping that in mind too, you don't want it too hot. You don't want it above really, 28 or 30 degrees. I wouldn't like to get it above at all. Even 25, I wouldn't like to get above. So that's like 78 Fahrenheit, and then just get a dehumidifier. That works perfect. You just put a number in. So if you've got a nice dehumidifier with you can dial a number into it. I used to put it on 55 for the first 2 days, and then for the next 10 days, I'd leave it on 60. So it would slowly get down, because the buds were they were 4 fingers wide. If you put your fingers behind a bud and look at it, you can count how many fingers, your index finger. So just go index, middle, and then it goes right to your pinky. That's the buds that I used to grow, the 4 fingers wide ones, mostly, not all the time, of course. There was always not perfect runs. So that means I'd have to slow dry it to get the moisture out of the middle of it properly. So it would, so when I did go to the cure, it would maintain the terpene profile that was on the plant. It wasn't too moist to start and break it down, to react with the water and the oxygen that's left in there. All right, that was a pretty good example. Let's see what questions are on that. There's none. All right. Well, I hope that helped. I'll get back to some microbe stuff, because that was just really the Rhizophagy I was talking about. But there's a lot more microbe fun stuff. I'm not on the right screen. Hang on, just bear with me. I'll just go back to the microbe screen. Microbes. Let's see what we can talk about in here. Microbes are fun. They're just so needed. They're so beneficial. What's some cool stuff? Microbes availability of nutrients. Oh, that was that thing before. So the availability of nutrients for microbes. So they always need nitrogen for their amino acids and enzyme production. They always need their phosphorus for their energy and their DNA. And they always need their sulfur for coenzymes, calcium and magnesium. So see, by having microbes, and even if they're dead, you're still going to get your NPKs that are into the plant's nutritional elements requirements, because it needs 17 of the plant, the cannabis plant, and the first 3 are CH and O, carbon, hydrogen, oxygen, which is 95%. And then the next one is nitrogen. Actually, I'll just show you, because it's pretty cool. It's a good, you can work out what your plant needs by knowing what it wants. Where is this going to be found? I better stop. And then I'll look for it. It's pretty essential though. So it'd be good to talk about it. Plant science, it should be. Yes, there it is, right there. Oh no, I'll get the one with actually the better chart on it. That one. Is that the latest? It doesn't matter, I'll just still talk about that. It's close enough. Actually, no, it's not close enough. I'll explain it while showing. How's that, so you know. This one. So you got the first 3 of a plant, essential elements, are CH and O, which make up 95%. It's 96, it's actually a 95. Because the science was always changing, this is why science is rad. Because different processes get experimented on, different outcomes and experiments are proven and disproven, and the essential elements changed. From 19, it made the silicon and chlorine as mobile elements, meaning that they're not an essential for all plants. And for medical cannabis, I'm not sure if chlorine's too essential. So I just talk about 17. I did a course last year. It was written last year, and that's where they had updated information on this chart, which I should probably be showing. But anyway, the next one's down. It goes from nitrogen, 1.5% is all that is required of a plant out of 100% to grow. And then there's potassium, 1, calcium's 1.5%, magnesium's 0.2%, then it goes phosphorus, then sulfur. And then it goes into your ppms. So they're so low of an element that they're into the micronutrients they're scaled, classified as. But you can see up here, all of these come out of microbes. So if you've got microbes in your soil and they're broken down, the NPK structure of most microbes are what is essential for plants to grow. That's what the whole point in showing that chart was. Over here. So just by having them, it really works out good. In general, microbial growth in subsoils is not limited to nitrogen and phosphorus as long as the contaminant concentrations are in the subparts per million. A lot of phosphorus is not used each grow, and it's in the soils, it's 45%, I remember, that's left in the substrates that the plant can reuse next time, because phosphorus is bound up in DNA. That's the major organic nutrient supply of it. So all of those microbes that are unused or unbroken down, they're still mineralized. They're still immobilized. They haven't been mineralized and broken down into the mineral form yet. They are still available. And the carbon, nitrogen, and phosphorus ratio of 30 to 5:1 is generally sufficient to ensure unrestricted growth in aquifers. Oh, okay. Very good. Better read this before I trace elements like that. Okay. In general, okay. What else was there? Don't want to know that one. This is a good chart. This isn't shown really much. This is the pH level of the primary and some secondary nutrients, but it also includes fungi and bacteria. So you can see fungi is prevalent throughout the pH scale, but bacteria, it really dwindles in the acidic scale, especially when it goes between 5.5 and tapers down to 4, it's nearly gone. There's really only special types of bacteria that specialize in those acidic profiles. I like that chart. Serial dilution. I like that because of the artwork. Pretty cool. Identifying microbes is to do with their morphogenic shape. And pretty much by looking at their shape, you can see these ones, the round ones are coccus. The long skinny ones are of a bacillus or a pseudomonas. So you can really get into them. Then the helical ones are the spiral ones. Oh, isn't that funny? And here it is. So just how to identify it. It's like as if I just read it. I didn't know. The vibro ones are the banana-shaped ones. I remember seeing some of them under my microscope. I've got a cool compound microscope I should pull out and put some live samples under it, and you can see everything moving. So you can see all the different types of shapes. There's a spirillum and spirochetes, fusiform bacillus. Then you can identify bits and see what's in your soils or your leachette or in your worm castings. It's pretty cool to see how active a lot of these things are. This is a plate just to see if I proved some species biological control. It's really good. It's really good, yeah. It helps a lot. It's a must, really. That's major processes of the nitrogen cycle. No, don't get into those. This explains microbes at their different. I don't know if this was really relevant to medical cannabis. It's more so environmental. It's just how they mineralize and they can leach things, they oxidize things, they reduce things, they absorb stuff, they accumulate things. So the different types of microbes that go through these different processes. Because only 99, there's only under 1% of microbes have been cultured and categorized. tDNA. This is a cool, where a microbe called Agrobacterium species. It's transfers genes, TI plasma genes, from one cell to another cell. And it's one of the first ways that they did gene manipulation in plants. Through, you'll see on the plants, they have this big crown gall, it's called. It's a disease, crown gall disease, and that's got the agrobacterium species inside of it. And that's what these are capable of doing. It's pretty fascinating. Contaminants can serve as electron donors. Okay. The ability of electron acceptors. That's how microbes work. They need an electron acceptor and electron donor. Exercise of organisms which require degradation. No. All right. Let's get to one that we know. Here we go. So nitrosomus transforms NH4 to NO2. Ammonium it transforms to nitrite. And then nitrobacter transforms NO2 to NO3, which is available to plants. So by having them, they need an electron acceptor and electron donor. So they'll have their oxygen is their electron acceptor, and then their electron donor will be what they're transforming it into. And that's how they function, I suppose. That'll do. It gets too heavily into it. This is how to, this is a bit, actually, YouTube doesn't like it. Alternative methods for isolations of pure cultures. This is cool. This is very cool. You can culture our azos from the air. Azotobacter are a beneficial biostimulant, like a biofertilizer even. They fix nitrogen non-symbiotically. So they work extremely well with medical cannabis. And you're going, well, how can I get some? You know, that's just unavailable to me. Well, you can get them. Just you have to grab some soil, inoculate it, plate it, cereal dilute it, and then you're left with the ones that will be fixing the nitrogen in there because there's none of the ammonium put in there that it fixes. And then you'll go, cool. Then you can work with it. Isolation of pure cultures, specific microbial methods, microbial communities from environmental systems. No, that's called a metagenomic study. Metabolic. No, that's to explain something different. But a metagenomic study is when you study all the microbes in a community. 6 categories. Yeah, this is what's happened in the world. These are so far what they've cultured in the world. So 0.1% of sea water bacteria has been cultured. That's it. That's it. But activated sludge, like in waste treatment plants, has been quite a lot. Between 1 and 15% has been cultured, because that's where a lot of studies has been done to work out how to reduce that material so we can have not a build-up of crap everywhere. So, yeah, it's pretty cool. Microbes are rad. So preparation of soil dilution. Oh, yeah. You put in 100, take out 9. Actually, I'll see if there's any following figures. That's pretty cool, how they break down. See if there's any questions on microbes. This is pretty cool, microbes. Hey, martial artist, how's it going? Martial artist, nice to see ya. I might smoke one to celebrate. Very good. Oh, what? This bloke's in the house, hey? Main green grower. There you go, mate. Plants are taken off in the 65 gals. You beauty. I hope it does well, mate. 65 gal pots is the go. No till preferred, he says. always that. So that maintains the plant microbe relationship. Having no-till, and then you can amend a lot of people think, oh, but I can't get the things I need out of it. I can't get the yield or what I want out of it. Well, you can amend it, but you'll get a better secondary metabolite profile. That's how you win comps and win medicinal things. Microbes, plant relationship. I have to try and keep focused on that topic. Don't want to get sidetracked. Yeah, this is pretty cool. The metabolism. Out of your metabolism, it's made up of two things, your catabolism and your anabolism. And the first one, the anabolism makes it and reduces the power and uses it. Hang on. Synthesis of material, reduced molecules, and oxidation of reduced substances. Catabolism, the use of substrate level phosphorylation, electron transport, so that makes it, and biosynthesis, that uses it. So assimilation, reduction. So yeah, your catabolism makes the power, your ATP, and your anabolism uses the power. Oh, and that's what it says down the bottom. Okay. All right. But yeah, just the metabolism of anything, of humans as well, any cells. Rapid extension periods of rapid microbial growth in natural habitats. Yes, if you get the microbe in 48 hours, a single bacterium exponentially doubling its biomass every 20 minutes would produce progeny that roughly covers 4,000 times the weight of the Earth. So it's so hard to believe. If you get an E. coli, put it in the right conditions, it would double. So you got it here, 20 minutes later, it's got two of them. 20 minutes later, two more. 20 minutes later, after an hour, it's got all these. And it keeps on going out and exponentially goes out. This is the bar graph. It just keeps going up. Yeah. So that's, and it's more so to do with the activity water level. I should bring that chart up. I will. Yeah, these are all the 5 major nutritional types of organisms. Chemo, synthetic, so chemicals and synthetics. Photos, the autos, photo is the sun, and synthetics. Chemo, where's lithotroph then? Oh, it's over in this one. Litho's rock. Heterotrophs means both. Let's get to that. Guidelines. This is what is very important too. I did a course called Foods Microbial Practices or something along those lines, Food and Safe Microbial Practices. And it was, a lot of it was on this sanitation, disinfection, and sterilisation, the difference in them all. And the reason why it's good to know these is because you can know how to treat your microbes. Because really we're trichome and terpene and flavonoid farmers, and inevitably, but to do that, the plant's got to house those, and for the plant to grow, it wants a microbe interaction massively. So it comes back to learning how the microbes work. So by learning what disinfects them and kills them, really, and it's good to not put any of those types of materials in your compost or any of your practices. So like soaps, greases, alcohols, bleaches, bad, it sort of speaks for itself, but that's the whole reason for learning that. I thought it was quite cool. This is pretty rad. This is to know the stratified layers of oxygen in the soil particle. So if your soil particle is more than half an inch wide or 12 millimeters wide, I always bust them with my fingers because there's a chance that it could have 0 oxygen inside of it, meaning it could be culturing all your Umiceti pathogens or any other pathogens that are problematic. So this is a good, Brock's Book of Biology. I really support that book. It's rad as. There's a 15th edition now. It's just something now. It's just really good. I love opening it up. the resources that govern microbial growth. So yeah, it's because we are in medical cannabis, we want the conditions to be around the room temperature, which is 24 Celsius, or what's that in Fahrenheit? 75 odd, to maintain the microbes that we want, and then they'll culture and live symbiotically with plants without breaking this down. Because yeah, they change. This is for all microbes, so it goes right into fresh water, salt water, all sorts of different conditions and oxygen levels, pH levels. I'm going from the back again. That was a really good one, that one. I'll go from the start, so reading the other way. Oh, that's pathogens. This is the plant relationship, soil relationship with the microbes. So you can see the blue ones are are bacteria, and the red ones are the fungi. So in all different tissues of the plant, there's all different types of species and genuses of bacteria and fungi, all sorts. I'm not going to attempt to name, there's hundreds. Enterobacter I didn't say before either. That's one that's I use, which is quite good too. Yeah, everywhere in the plant. Very, very beneficial. All right. This is a cool little experiment. This is an assignment I did for a month just to see the phases of microbial growth in a tease. So, yeah, that's how they should go. They got lag and then log, which goes up after a few hours. Then it flattens out when they've run out of their electron acceptors that they want to continue their growth. And they run out of things, then they start to die. So that's why you want to get microbes in the right phases. That's why I thought, I wonder what phases my teas are at. So I did quite a few different plots at different stages to see how they were going and to see if I'd run out of any of their nutritional requirements. So they're in their death phase. And one of my microbial professors, she did her doctorate on this, and she kept one culture going for 9 months, just flatlining. And see, look, even with the root tea, it even went up at the end after 2 days. So they would have, so something else positively influenced them. So different processes happened for that to happen. Yeah, it's pretty cool. Good fun understanding microbes. And plants have, yeah, 2 immune systems, both reliant on microbes. Oh, really? It's a systemic acquired immunity or a systemic innate immunity. So one's induced and one's non-induced. Cut a long story short. Innate immunity is activated by rhizobacteria, which are recruited from the soil microbe community. So if you've got your beneficials down there, then it's going to activate it anyway for you and do it. Different pathogens, BTAs. Okay. That was, that's how to do it. Plant microbe association. Wow, plants are plant tissue general, depending on the impact of microbes on plants, they can be categorized as a pathogen, a plant growth promoting bacteria, or a commensal. So plants depend on their microbial partners for many functions. Soil microbes, so beneficial. Up to 1 million cells of fungi can be obtained out of 1 gram of soil. And also 1 billion viruses. Yes, healthy soil has equal amounts of pathogens to it does as beneficials. And because there's so many beneficials in soil health keeping in check, that they keep a healthy profile. I learnt that last year with my soil science class, the new one. the phytomicrobiome is the community of all microbes that are associated with the plants. Oh, I'm not going to read all them. Rhizoplane, rhizomite. Yeah, good. The rhizosphere is the area around the root. The rhizoplane is the area attached to the root. That's a good summary of all that. Plant microbe relationships. Yeah, so that was the Rhizophagy cycle, which was showing this process here on the endophytes. Went through all that. Plant growth promoting bacteria. A well-structured, regulated and complex community of microbes. They're so beneficial, and there's so many good ones in there. They give hormones and all sorts. So it really pays to have a good diversity of microbes. They colonise the root space and the plant material as well. They were introduced by Kloper and Schroth in 1980 and labelled PGPR, plant growth promoting rhizobacteria. Extracellular, so living on the outside, so they would be in the rhizosphere, not inside. So they are ones like azos, like I talked about before, and the bacillus and the pseudomonas, very beneficial to medical cannabis. And the intracellular, what about them? They live inside. So they're the endos, and they specialised nodular structures, okay, symbiotic relationships. Yes, and they're ones like the Rhizophagus irregularis and the bacillus and some pseudomonas species. Plant exudates, plant hormones. Yeah, because the plant puts out in the rhizosphere, the plant puts out organic acids or hydrogen protons, and that exchanges with microbes in their needs, because they need the hydrogen for most of their cellular practices, like I shown before. No, it's not up there. So they can continue that and they'll exchange it. So they'll grab one and then they'll swap it and they'll give the plant roots back another one. That's how the plant gets its root exudates. Plant secretes several molecules that's specifically to microbial growth and activity. So they try and attract them because they want to get the ones that they're going to be symbiotic with. And the root exudate comprises of its organic compounds like sugars, amino acids, organic acids, secondary metabolites like flavonoids and terpenes to attract the microbes that it wants. And they also have a high molecular weight and have proteins and polysaccharides. They attract specific microbes in the rhizosphere, directly relate to microbial interactions. They can mediate both positive. The root exudates can mediate both positive and negative interactions. So the positive actions, like symbiosis, would be mycorrhiza, so the fungi working with around in the area, or the plant growth promoting rhizobacteria that's working there too. The negative interactions would be like the parasites and the allelopathy, where the plant speaks to each other and they exchange chemicals that way, and competition. And the phytohormones, so many of the microbes, they put out the hormones in the plants just naturally, and like gibberellins, one of the gibberellic acid, it comes from, it's gibberellins, fibrosinins. Sorry, I can't remember the pronunciation, but there's a microbe that produces gibberellic acid. And other ones do it as produce different types of plant hormones as well. So it's what this is basically explaining. They help a lot. The phytohormones, yeah, they just help. I'm not going to go through all that. They produce cytokines, yes, another form. Auxins as well. Ethylene, all of the beneficial ones. Other signals. So Plant growth promoting rhizobacteria secretes several secondary metabolites and volatile organic compounds that improve stress tolerance and stimulate growth, like Bacillus magnetarium. I think magnetarium should be what else? Microbes. All right, what else is there? No, probably not about that stuff. Direct plant growth promotion. So microbes help in direct plant health promotion by facilitating resource acquisitions like nitrogen fixation, phosphate solubilization, siderophores, which like chelate things, and sequestering iron. And another direct plant growth promotion is the hormones. Some indirect plant promotions, though, they're antibiotic productions, because microbes want to have it all to themselves. So they put out that biofilm, which is an antibiotic, and it repels microbes. That's why some, or quite a lot of microbes aren't symbiotic, particularly lactobacillus. Those species are not real symbiotic. That'll be facilitating resource acquisitions, like nitrogen. Yes. It wants to facilitate it all. Yes, it does. It needs to produce nitrogenase to break it down. Involved with the NIF gene. Non-symbiotic nitrogen fixation. Those are the azos. So that's the ones that we want in medical cannabis. So they're performed by free-living diazotrophs, which they fix atmospheric nitrogen gases into ammonia. And then they transfer it from that processes, which nitrosomes, and then through nitrobacter into the NO3, which plant requires. And then there's two forms of nitrogen that the plant is suitable for, the ammonium, NH4, and the NO3, which is the ammonia, NO3, which is the nitrate. Diazotropic bacterial species have been cultured for at least 50 genera. However, the numbers are much more. Based upon their physiology, these are anaerobes. No, we want to know about the aerobic ones, because that's the only ones that are more so involved with the cannabis. These ones, cyanobacteria, are formerly known as blue-green algae, but because they've got a prokaryotic, a round bean-shaped cell, they're a bacteria. They are beneficial too, but I use them. Do you know that they're 70% tryptophan? Non-symbiotic nitrogen fixation. Symbiotic nitrogen, no, because we want non-symbiotic because it doesn't nodulate. Phosphate solubilizing bacteria are extremely important, like Bacillus. They're very good at doing that. And some other types of ones. It doesn't say. But they transform and break down from an immobilized form of, so if you've got other bugs or something that's holding up phosphate inside of it, it's going to reduce it down into an available form in the plants. So if it's taking it from a immobilized into a mobilized, mineralized version, which is available. So they mineralize it. Yes, that's what we'll do for that. PGPR synthesizes phytohormones like auxins, cytokines, etc. That'd be good. We should go through all the hormones ones. That's a good one, actually. PGPR produces auxins. Indole abscisic acid are well studied, as auxins stimulate root branching and elongation. They increase, this goes into the hormones, which I'm not going to get into. And they also may regulate the size of stomata and their density. Isn't that interesting? Righto. Siderophores, no. Indirect mechanisms. Mechanisms of action. Righto. Come on, summary. Mechanisms of actions of plant growth promoting rhizobacteria are nitrogen fixations, phosphate solubilizations, and chelating. And indirect mechanisms involve enzymes, antibiotics, and systematic resistances. So they're quite beneficial. Oh my goodness, it's been over an hour. See, I just love microbes, dead set. Where was up to? Blinky. Oh yeah, oh that's good. 13 watching, thank you. And no-till, top dress. Bam, fire, yes. That's a microbe's way of doing it. No-till, top dress, tea, and again, bam, fire, bang. That's the go. And it's amazing how well it works. You've got to put so much effort into the substrates where basically takes care of itself, when other way of growing is putting so much effort into taking care of all the pesticides, fungicides, herbicides that you need and deficiencies that the plant gets, because the plant's not in its natural form. It's tried to be manipulated with chemicals. So big, massive difference. I'm so for that way of, on the screen, of growing agriculture, medical cannabis. Keep up the good work. Blast it. Yes. Thanks. Oh, yep. Thank you. We'll catch up and when get home. Cheers. What? Okay. Odd. But it's the way of growing is with the microbes for sure. What else? There's no been real. I can't believe the time. I love talking about microbes. Cool. So I can change the title of today of the microbes and drying, curing, I suppose. But it was really just on the microbes. There'd be more stuff I could show on microbes too, I bet you, if I tried a bit. Actually, it's been going long enough, hasn't it? Yes, you say. Yes. Protozoa. What's that one? Oh yeah, this is even a different one. A chart just to wrap up with microbes. I was showing before with their NPK breakdown. This is another one. Their cell chemistry and nutrition. So it's basically the same one that was before, but it's just written a different way. And you can also see in here, it's got their moisture ratings that they need. So that's their activity water level. So if you maintain that, that's what they want. They will facilitate, because see in their prokaryotic cell, if you want to break down and look inside of it, half of it has a endospore and the other half is their organelles to function. So when they die, the other half in the cell creates that spore-like thing, or a seed like this. Even though that's a fungal one. So it's a little smidgen different from bacterial one, but that's how they reproduce. So it's so easy for them to continue on. That's why if you get a lot of sewage sludge or manures, they have a lot of these in them as seed form. So it's a good way to get your microbial diversity up. You think, oh, they're all dead in there. They might be dead, yeah, but given the right conditions, they will easily come alive and proliferate after that, multiply their cells. And another one, the macromolecular composition of a cell. It just shows in here that it's got 55% of it is actually protein inside of it. 9% lipids, and it goes down there. 3% is DNA. So out of that DNA, you need, to make up DNA chains, there's a lot of phosphorus in there. So you can nearly say that it's 3% is phosphorus. Actually, it's not that high because there's other different compounds to make up DNA, but it's high anyway. Yes, cool as. All right. Well, next week I'll see if I can get on to Frank and see if I can put on the breeding one and talk about that stuff again. And that'll be good. That'll be another big, long show, I reckon, too, because I get excited about breeding, just like microbes. It's fun. It's just an experiment, and you can experiment and do it yourself and see what happens. You're not hurting anybody. And generally you'll get a good outcome. A couple of little pointers in the right direction, a few bits of microbes. Yes, I'll try and do that. Otherwise, I will be posted as this one, an open topic, if I can't get on to Frank. Yeah, and I hope to see everybody next week. Thank you for coming and sitting and listening and standing or whatever you listen. And I'll catch you next week. So happy breeding, happy growing, and good health to you all. Thank you. Bye-bye.